Hi everyone, my name is Marina, and today I'll be presenting my master's thesis titled Hold the Salt, Are There Spatial Bulk Health Refugia from Purple Sea Urchins Near River Mouths? And this is in Mendocino County. And so I'm testing to see if the decreased salinity in these areas near river mouths are negatively impacting purple sea urchins, which allows for bulk kelp to thrive in these areas. Oops. So I want to start off with a land acknowledgement. And so I go to school at Cal Poly Humboldt, or formerly known as Humboldt State University, which is on Wiyot land. And I conducted my laboratory experiments in Trinidad at the Marine Lab, which is home to the Trinidad Rancheria. And then my field observations were done in Mendocino County, home to the Sherwood Valley Band of Pomo Indians. And then a little outline for today. So I'm going to do a little background on kelp and urchins and in relation to salinity. And then I'll go over my hypothesis and how I'll answer those with field and laboratory methods. And then the results of those field observations and laboratory experiments and the biological implications from those. So I'm sure we're all very familiar with these figures, but they're very nice for presentations. So since the mid-2010s, we had a major loss in bull kelp canopy coverage in the North Coast. And so on the far right, you can see a lack of that green in Mendocino and Sonoma County. And this is due to a series of events that created this sort of perfect storm. So from around 2014 to 2016, we had these warm water anomalies that increased sea surface temperature. We also saw sea star wasting disease affect about 10 different species of sea stars, most notably, or one of the most notable, as Pycnopodia helianthoides, the sunflower sea star, which is a known predator of purple sea urchins. And then we saw this population increase of purple sea urchins as well. And then we saw this decrease in our red abalone population, ultimately closing the fishery in 2018. And I also want to note that we saw a change in urchin behavior from this passive feeding behavior to an active feeding behavior with the lack of predators present. And so we had this loss in bulk kelp from this increase in temperature, but then we've had a sustained loss with purple sea urchins continuing to eat any bulk kelp from coming back. And so urchins, they're a type of echinoderm, and so basically they use a water vascular system, meaning that they're particularly sensitive to changes in seawater chemistry, such as changes in salinity. And so basically what that means is they take the seawater surrounding them and they pump it through themselves internally for physiological function. So with urchins, they have these little tube feet for hydrostatic adhesion. And so when you change the seawater chemistry, that can disrupt that, where they use their tube feet for locomotion and foraging. But then if you go low enough, and in some instances, or there has been recorded instances of mass die-offs of purple sea urchins associated with large freshwater input in this case Malibu in 2013. And there's been other studies that have recorded this in recently that has tested salinities for other species of urchins. Although there is one species of urchin, the green sea urchin, that is known to tolerate low salinities, so we can't just necessarily assume that all echinoderms, or in this case all urchins, will react the same way. The green or sea urchin is also particularly special because it's a circumpolar species, meaning it's found across the Arctic and all the way on the East Coast as well. And then bull kelp can survive or has some survivorship down to low salinities as well, even as low as 11 PSU, although it does have some negative effects. And so this is really interesting information because folks have had some anecdotal observations of persisting kelp since that mass loss in bulk kelp during the mid 2010s, particularly at the mouths of rivers or freshwater input. And so I took this screenshot from Kelp Watch on the far right over here, excuse me, which is at the mouth of the Big River or located at Portuguese Beach in the town of Mendocino. And the same creators of Kelp Watch also came out with a publication last year that identified Mendocino as a kelp climate refuge. And so basically, we're seeing these persisting kelp beds at the mouths of rivers, and I'm hypothesizing that this is due to urchins being echinoderms and not being able to tolerate these changes in seawater chemistry. And so I'll test this with a series of laboratory experiments and field observations, or I did test these, excuse me. And so I asked the following questions, starting off with the lab. If decreases in salinity negatively impact purple sea urchin grazing on bulk kelp, if intermittent pulses of these decreased salinities impact urchin grazing on bulk kelp, and if these decreases in salinity impact urchin mortality. And then for the field observations, I'm going to measure if purple sea urchin populations differ at river mouths compared to non-rivering sites. And then I'll also measure, excuse me, if the purple sea urchin population size distributions differ between river mouths and non-rivering sites. And so I'll start off with the field observations. So on the far right here is my field sites. So river sites are the blue pins and their paired reference sites are the red pins. And so I did Noyo River and its reference site Hare Creek, the Big River with its reference site Mendocino Headlands, also known as Pipeline, the Albion River and its reference site Dark Gulch, and the Navarre River with its reference site Saddle Point. And then on the bottom left here is a photo of me and my lab partner, Duncan Jackson. And then what I did out in the field is I randomly placed 8 transects at a site either in one of these persistent kelp beds or around a deployed CTD logger that we installed to measure conductivity, temperature, and depth. And so with these transects, we counted urchins and kelp, and then also did urchin sizing. And with these counts, we not only did purple sea urchins and bull kelp, but other organisms that we believe could influence kelp and urchin abundances. So this is like red urchins or other species of laminaries that could outcompete our target species. And then for sizing, we sized up to 30 urchins per species for reds and purples per transect to the nearest centimeter. And our CTD loggers take readings every 6 minutes. And here's some data from one of those loggers. So this is at the Albion River. And so we installed this logger back in August of 2023. And we had it over winter, but we ideally would change out these loggers every 3 months to avoid drift. But if you've dove the North Coast, you know it's a little bit difficult sometimes, and it's weather dependent. So we're going to try for a second winter season coming this year, and hopefully we'll have, or we do have more data, but hopefully we'll have more winter data to show. And so on the top here we have temperature, and on the bottom we have salinity. And I added in these 2 red bars that go across, one at 25 PSU and one at 20 PSU. So it's easier to see that salinity does drop below 25 PSU. And this is just with a couple of small rain events. And so before we were really seeing the winter season come through. So I do suspect that salinity will be dropping below that 20 PSU most certainly during the winter season. And then getting into the results of the field observations. So on the left here, we have adult bulk kelp per individuals per meter squared. And again, our river sites are in blue and reference sites are in red. And what I found was that there was no statistical difference between river and reference sites for bull kelp individuals per meter squared. And then on the right, I have purple sea urchins per meter squared. And you can see here that we have little to almost, some sites have no purple sea urchins present. And then at the rivering sites and then at the reference sites, we have hundreds of purple sea urchins per meter squared. And I did find a statistical difference for purple sea urchin density between river sites and non-rivering sites. And then this is the data for all sites for all species. Or all site pairs, I should say. So on the top, we have Noyo River and Hare Creek. The second one down is Big River and Mendocino Headlands/Pipeline. The third one down is Albion River and Dark Gulch. And then the fourth one down is Navarro and Saddle Point. And the only thing missing from here is purple sea urchins because the scale was so large that you wouldn't be able to see the other species. And here I have some observations that I made based on these graphs where Noyo River and Albion River were inverse to their reference sites, where the rivering sites had more algae abundance and the reference sites had more invertebrate abundance. Whereas Big River and Navarro and their reference sites, Mendocino Headlands and Saddle Point, had more broad distribution or a mix of both kelp and or algae and invertebrates. Let's see. And then getting into the size class distribution. So once again, there were almost no urchins present at the river sites, or there were very few at Big River and Navarro and a single urchin at Albion. And in this case, I am determining recruitment to these sites as the 1 to 2 centimeter size class. And so that's based on a previous study that indicates they're about 6 months to a year old. And so we can see that because there's almost no urchins, and especially no 1 to 2 centimeter urchins, there's either no recruitment happening at these sites or there's high rates of post-settlement mortality. Whereas for our reference sites, they all had some 1 to 2 centimeter purple sea urchins, and they all had a mix of reds and purples, but almost no 1 to 2 centimeter red urchins, with the exception of Dark Gulch. And in particular, at Mendocino Headlands/Pipeline and then Hare Creek as well, they had over 40 and almost 60 1 to 2 centimeter urchins, which indicates that there is definitely recruitment happening at these sites for purples. And then getting into my laboratory questions, my urchins have drifted a bit on my bullet points, but that's okay. Just to remind you guys, I'm looking to see if decreased salinities negatively impact urchin grazing, if intermittent pulses of these salinities impact urchin grazing, and if these decreased salinities impact urchin mortality. So I designed two different kinds of lab experiments here. One was just a constant exposure to salinity for a given amount of time. So I had between 6 and 9 replicates where I had one urchin in a tank sitting in a water bath where it did not have recirculating water outside of its own tank. And I tested salinities 32 to 10 PSU. And my control for this is kelp without an urchin to measure changes in mass unrelated to grazing. All urchins were starved 24 hours before the experiment, and I ran this for 72 hours and took one measurement every 24 hours. And I measured my kelp with wet weight, so I just blotted it with a paper towel and weighed in grams. And my formula to adjust for control is down here at the bottom. And then these are the results of that constant exposure to to decreased salinities, which I've named my press. And here I have graphed on the far left my 24 hours, in the middle my 48, and on the far right my 72 hours. And I put in some black X's where after 24 hours, all urchins at 18 PSU and lower were found dead. And then at 48 hours, I had two more urchins in 20 PSU found dead. And then in 72 hours, I had another 20 PSU urchin that I found that it was dead. And so we do see this relationship where there is a increase in urchin grazing with increase in salinity, but that might be due to the fact that the urchins at the lower salinities did not survive as well. But it's also interesting because there seems to be a threshold at around 26 PSU where the urchins are consuming more. And I did also have some interesting behavioral observations where I had two urchins in 22 PSU and another urchin in 24 PSU that was not actively feeding, or my urchins in those salinities were not feeding at all, but in particular, I had a couple of urchins trying to actively escape their tanks. And I noticed that at 24 hours, and I also noticed them repeating that same behavior by 72 hours as well. And then I also ran another laboratory experiment, and so this was that intermittent pulsing. So this one I did about half the amount of salinities due to capacity, so from 32 to 12 PSU. Um, basically, how do I say this? Like in increments of four, I think. Yeah. And I had 6 to 8 replicates per salinity. And so the way how I did this lab experiment is I took a baseline for 24 hours where I just had them at regular ambient salinity. In this case, that's 32 PSU. And I measured once every 6 hours. And then I started pulsing them every 6 hours by doing a manual water change. And so you can kind of see this like stepwise function here where the example drops to 12 PSU. And then I switch out the water, bring it back to 32. And so I did that. Um, I changed seawater every 6 hours for 5 days. And then I left them at 32 PSU and measured for 6 hours for one more day, and then left them in this sort of recovery period at 32 PSU for 7 days after. And so, similarly, with the constant exposure to decreased salinities, my control was kelp without an urchin. Urchins were starved 24 hours before the start of the experiment. And what was different about this experiment is that I put in a new piece of kelp every 6 hours with those water changes to try and accommodate for osmosis, where the kelp will absorb some water with the decrease in salinity. And then I again, did a wet weight measurement where I blotted dry of paper towels and weighed in grams and used the same adjustment for control equation. And then this is the results of that lab experiment with the intermittent pulses. So up at the top here, I have my 12, 16, and 20 PSU urchins, and at the bottom we have 24, 28, and 32 PSU urchins. And overall, we can see that with those higher 3 salinities, our urchins ate a lot more. But that might be because the 12 and 16, and along with one of the 20 PSU urchins, died during this experiment. And I haven't graphed when urchins died for the intermittent pulses of salinity experiment yet, but the 12 PSU urchins died first, then the 16, and then one of these 20 PSU urchins later on. And then I also, oh, and so um, my conclusions overall from the questions I asked earlier. So for my field observations, I would say yes, sea urchin populations do differ at river mouths versus non-rivering sites. There were significantly higher abundances at the reference sites compared to the river sites, where there were almost no urchins or no urchins at all. And then I would also say that the population size distributions also differ because there are no urchins at these river mouths, whereas so then they're not there's either recruitment is not occurring or they're experiencing high rates of post-settlement mortality, whereas the reference sites have large abundances of urchins, and even some sites have large abundances of 1 to 2 centimeter size class urchins, indicating that recruitment is happening at some of those sites as well. And then for the laboratory experiments, if do decreases in salinity impact urchin grazing on bull kelp, I would say yes. With lower salinities, urchins consumed less kelp biomass, and I would say the same for intermittent pulses of decreased salinities, that we saw less consumption of kelp with decreased salinities. And then do these decreased salinities affect urchin mortality? I would say yes. With lower salinities, you have increased urchin mortality for both the constant exposure to salinity and the intermittent pulses of salinity. And this slide was supposed to go before the last one, but this was more physical characteristics that I recorded. And so I have here on the far left, detached tube feet. The second to the left is an urchin that's spawning. The or third to the left, or two to the right, is an urchin where the fleshy area around its mouthpiece, the Aristotle's lantern, is enlarged. And then the urchin on the far right is a dead urchin. And so the biological implications from this, or like the big takeaway picture, is that this will contribute to my field by informing our knowledge and understanding of what's like occurring at these climate resistant locations in order to help better inform restoration efforts for managers and maybe prioritize these sites that have these freshwater inputs to establish kelp refuges. And his study also enforces previous studies that include more, by including more salinity values with the CTD logger, the effects of salinity pulses and urchin density at river sites and size distribution and mortality with a broad range of salinities. And it sets the stage for future studies that may ask questions with the results of my thesis, like do urchins avoid rivers or do they just die, for example? And then some other next steps to investigate. I know my thesis just focuses on salinity, but that is obviously nature. Everything's a covariable. And so something I noticed at my sites was differences in substrate type between the rivering sites and the non-rivering sites. And so that's something that I would love to investigate for a PhD, is also looking at the effects of sediment on purple sea urchin grazing with bull kelp. And so big thank yous to everyone who has been a part of this. Thank you to NSF, my funder, and collaborators at Davis and Santa Cruz, and all the support over the years, and my undergrad assistants and lab partners, and my dog Momo. And there's my email up there as well if you want to email me. And I see this is a recycled slide from WSN, but thank you to Kelp Culture for having me as well.